Hey everyone, welcome back to MK Tutorials. Today we are going to learn about GenBank and how to access GenBank. So basically GenBank is a database where you are able to find all the nucleotide sequences and you are able to retrieve all the nucleotide sequences, whether it is from RNA, DNA or protein. And how to interpret the nucleotide sequence in this database. So let us start. So first simply what you have to do is go on the Chrome. After that search for NCBI and this is the page that opens up in front of you. So I have explained this how to use this NCBI. You can watch on my channel. I'll paste the link in the description box. Let us start with the video. So firstly what you have to do is to find the nucleotide sequence you have to click here and you have to click on nucleotide. And let us take an example. For example, I'm taking insulin. After that, click on search. After clicking on search, you'll get so many examples here. So click on one example. For example, I'm clicking here. This is one of the example. Okay. Now, how to interpret this or how to read this thing? Now, if you can see here, यहाँ पे ऊपर GenBank लिख के आया है. That means this search result is from GenBank. If you can see here, यहाँ पर दिया हुआ है how to interpret this. So firstly if you can see here कि यहाँ पर locus दिया हुआ है, this is the locus of the insulin gene, and after that this is the length of the base pair, that is 1091 base pair, and the type sequence, that is mRNA, it is linear type, and this is the date on which it has been published. So यहाँ पर दिया हुआ है definition, that example of what is the name of the query that we have searched, that is glycine max insulin gene enhancer protein. This is the accession number. Accession number is the unique identifier number of the particular gene or protein. Then the database link here it is given, that is bio project, and यहाँ पे click करने के बाद you can search it more. यहाँ पे keywords दिए हुए हैं कि यहाँ पे हम क्या keywords search कर सकते हैं, तो हमको ये example मिलेगा. After that source here कि कौन से source से extract किया हुआ है. So that is extracted from soybean glycine max. The glycine max is an organism जिससे ये extract किया हुआ है and it is from soybean. उसके बाद यहाँ पे कमेंट सेक्शन दिया हुआ है, तो यहाँ पर मॉडल जहाँ से भी this record is predicted by the computational analysis, so and it was derived from this genomic sequence which was annotated using gene prediction method. After that if you can see here कि यहाँ पे उसके features दिए हुए हैं same as in comment section, जो भी चीज़ें दी हुई है, तो organism जहाँ से ये extract किया हुआ है, that is glycine max, the molecule type is mRNA. Then the cultivar who cultivated it, the name of that cultivar, then the reference from where it is taken. After that chromosome इसका unknown है. Tissue type it was extracted from callus type tissue. यहाँ पर gene की information दी हुई है. If you can see here, this is the information of gene. यहाँ पे gene ID भी दी हुई है. You can click here and you can go on another page, then you can search for the gene ID. And यहाँ पर जो है वो coding sequence दी हुई है. So this is the coding sequence. So this is how you predict it. You can click on FASTA and you can download the FASTA sequence and you can search for another things, like you can run blast, you can do more things, like you can search the protein structure, you can search for this gene structure in other databases. After that you can do one thing, that you can click on graphics and you can see more things like this. Now see, depending on your sequence it will take time to load. So this is the graphical representation of the glycine max insulin gene enhancer protein. This green line represents the DNA sequence and यहाँ पे उसकी information दी हुई है. This red line represents about the protein sequence, that यहाँ पे दिया हुआ है coding sequence, qualifier name of the protein, location where it is located, and this black line represents a domain of the protein, and this line represents about the binding site of the protein. Okay, so this is how you can search about the particular gene or protein by using GenBank. So this was all about how to use GenBank. If you have understood the video, just like it and share it. And if you're new to the channel, just hit the bell icon so whenever I post a new video, you will be notified about it. Okay, bye.